Is it visible?
It is visible, yes, and on presentation mode, so excellent. Thank you.
Thank you. As you are looking my name on the screen, my name is Garoma Desa from Animal Health Institute, Ethiopia. I will speak on participatory epidemiology, including abattoir surveillance of contagious bovine pleuropneumonia. As an outline, we have an introduction, status and the drivers of CBPP in Africa, what makes CBPP endemic in Africa, what are the drivers, principles, purposes, and approaches of participatory abattoir surveillance, role of participatory abattoir surveillance in control of CBPP, what are the major findings of participatory abattoir surveillance for CBPP, why participatory abattoir surveillance is important, and at the last, limitations of participatory abattoir surveillance and recommendations to improve these limitations. Finally, summary. These are my outlines. Abattoir surveillance is the systematic inspection of animals, carcasses, and organs at slaughterhouses to identify evidence of infectious, zoonotic, or production-limiting diseases. As you know, abattoir surveillance is the combination of postmortem and antemortem inspection. We will inspect the animals before slaughter, either at rest or on moving, and we can also conduct postmortem examination of the organs. Participatory abattoir surveillance, when we say participatory, it is to mean empowering the community, empowering the participants. The participants are maybe the herders, the butchermen, traders, and it is empowering them to participate in solving the animal health problem or animal diseases. So participatory abattoir surveillance is a method that combines participatory epidemiology techniques with abattoir-based inspection. There are different types or different techniques of participatory epidemiology, including interviewing, visualization, scoring, and ranking. So, participatory abattoir surveillance is the combination of these participatory epidemiology techniques or methods with antemortem inspection and the postmortem inspection to detect, confirm, and understand the occurrence of disease patterns in slaughter animals. Participatory epidemiology. What is participatory epidemiology? As I raised on my previous slide, participatory is empowering the community to participate in the problem solving. Epidemiology is a wide discipline. It studies about the distribution and the determinants of the disease. Distribution, in essence, it may be spatial distribution, where is the place more prevalent, where is the disease is more prevalent, and where is less prevalent. It also studies about the temporal distribution of diseases, at which months, at which season the disease is more prevalent. It studies this distribution and also the determinants of the disease. There are a lot of determinants, but generally classified or categorized into three: host determinant, agent determinant, and environment determinant. When we say host, the disease or the incidence of the disease is different from species to species. Some species are more resistant to the disease, and some of them are more susceptible. There is also a difference in period in the susceptibility against any disease. So even it is the disease incidence is different between male and the female, and also between different age groups. The disease incidence is also different. When we say agent determinant, it is the pathogenicity, virulence, and when we say the environment determinant, maybe the climate. Every place have no similar climate. Some areas, some places have hot climate, and some others have a cold climate, which which differently treat the incidence of the disease. So epidemiology studies about these two important components, then the output of the study of the epidemiology will be implemented or applied to control the disease of animals, or that means the output of the study of the spatial distribution, the temporal distribution, and the study of the determinants will be used to control the disease. So participatory epidemiology is a form of active surveillance. It is one of the active surveillance. There are different types of active surveillance. This is one of the part of active surveillance in which participatory approaches and the methods are used to explore the local knowledge of livestock keepers on animal diseases, on its epidemiology, and on the impact of the disease. In participatory abattoir surveillance, veterinarians or meat inspectors work together with butchers, traders, and the community representatives like key informants to share knowledge and interpret findings as far as the community are living closely with their animals. They know a lot about the disease pattern. They have a good indigenous knowledge about the disease. So participating them in solving the animal health problem will make us more achievable. Why developed participatory epidemiology? Because conventional surveys did not represent reality due to sampling and measurement biases like spatial bias, project bias, person bias, seasonal bias, and there are also another bias. When we say spatial biases, most of the time we prefer to visit or to conduct a research in accessible district, in accessible villages, while some villages difficult to reach are left unstudied, left unvisited, which will result in spatial biases. There is also a project bias. Most of the time, the areas with external input, with projects or with external resources have a priority to get or to gain the research host. We also have a person bias. In nature, we prefer to ask men than women, wealthy or rich man than the poor one, educated than non-educated. But the women and the poor and the uneducated peoples may have good knowledge about the disease. So impartiality when we approach to them, separating them, or only preferring the main rich and educated people will community will make person bias. Seasonal bias is also another bias that we encounter during conventional surveys. That means we prefer during easy season, for example, dry season have no a lot of challenges. It is easy to walk, it is easy to work. There is no mud, there is no wetness at the dry season. But different seasons, different times have different conditions for diseases. So participatory epidemiology was developed to improve the biases, encountered due to conventional surveys. These are the pictures showing the participatory epidemiology. There are proportional piling, transient walk, matrix scoring. We can use proportional piling to know from which district most CBPP infected animals are originated. We can also use transect walk involving an interview, visiting the areas where is the veterinary clinics, where is the abattoir to visit, to also evaluate where is the for example, vector transmitted diseases. Where is the place for vector transmitted disease? To identify the places, we can use transect walk. Matrix scoring also is one of the participatory epidemiology tool to identify which disease symptom is commonly related with a given disease, for example, with CBPP. Shortly, this picture shows the different types of participatory epidemiology. What is the status of CBPP in Africa? My previous presenters addressed some of my topics. I hope this also addressed with somebody. According to World Organization for Animal Health, 22 African countries from West, East, and Central Africa were reported as the disease or CBPP is endemic in them, with only 4 countries officially reported as CBPP free, according to WOAH, World Organization for Animal Health. The 4 countries are South Africa, Botswana, Namibia, Swatini. The other countries reported as the disease is endemic in them. There is also recent seroprevalency studies revealing or revealed in a high seroprevalency. For example, in Uganda, in Sudan, and in pastoral regions of Ethiopia. Disease spread increasing in some areas due to drought-driving cattle movements and weak movement control. When drought happen in some area, there will be no enough animal feed. Then the animals and the communities will travel a long distance in search of animal feed and water. So this will increase the transmission of the disease. As we all know, there is weak movement control from place to place, from country to country, from even district to district. So these 2 important components increase the disease transmission. What are the drivers keeping CBPP endemic in Africa? Transhumans. Transhumans means in pastoral areas, the communities are known by long travel in search of food and in search of animal feed and water. They travel a long distance, a long kilometers, during which they transmit disease. They take disease from affected area to non-affected area. There is also porous borders. That means there is limited veterinary checkpoints to check or to test the animals. And there is, as I told on my previous slide, there is weak movement control. There is also low coverage and irregular vaccination and imperfect vaccines that make CBPP endemic in Africa, as my colleague or my friend raised on the opening speech. We have a problem of vaccine. There is a shortage of vaccine. In its nature, CBPP needs vaccine at least to give enough protection. We have to vaccinate our animals at least annually, or if possible, 2 times per year. But to keep or to maintain this regularity, we have no enough vaccine. So this results in low coverage of the vaccination and regular vaccination. And there is also imperfect vaccines. Sometimes we get or we feel post-vaccine reaction, which will decrease the trust of the community on us, and also this affect the effectiveness of the ongoing CBPP control program. This is another issue that keep the CBPP endemic in Africa. Also, it was
Dr.
raised by
Dr. Desa, I'm sorry. I am a bit mindful of the time. I think you should probably go less into details to make it in the next 5 minutes, to finalize in the next 5 minutes. Is that possible?
Okay, 5 minutes. Oh, I will try. Okay. The other drivers are use of antimicrobials in the field, surveillance gaps and limited resources, weak regional coordination. The key principles of participatory epidemiology. What are the key principles? In the participatory epidemiology, we have to think of our behavior and the attitude when we are working with the community. We have to respect them. It is good to listen more rather than to speak more. We act just like a facilitator, not as an experiment, not as an expert. It is also important to consider there is a co-learning, two-way. It is a two-way direction. We can learn from them; they can also learn from us. It is combination of the combination of scientific knowledge and the traditional knowledge, understanding existing knowledge, optimal ignorance, action-oriented. Optimal ignorance means, for example, it is no need to know each and every problems in detail. In order to solve major problems, it is important to know the some questions. So it is good to apply the principle of optimal ignorance for less important problems. Purposes of participatory abattoir surveillance: to detect CBP populations and confirm disease presence in slaughtered animals, to trace disease origin based on the animal source, to support the planning of targeted vaccination, targeted movement control, and targeted eradication programs. Approaches of participatory abattoir surveillance. The first approach is community engagement. We have to engage the community, which are butchermen, maybe traders, maybe herders, and others. We have to engage them, and also we have to train the team members, the butchermen, the traders, and the herders. We have to train them on how to detect, how to identify CBP populations at Abattoir, how to collect the data, how to interview, or totally how to use the participatory epidemiology tools. Then participate on the participatory data collection, ante-mortem inspection, post-mortem inspection, sample collection, multi-source triangulation. Multi-source triangulation means combining the results from community, the data that we get from the community, the data that we get from ante-mortem inspection, from post-mortem inspection, and from the laboratory result. Then we must triangulate, we must combine these findings if we are going to take action or if we are going to take decision. Trace back and trace forward activities. Trace back means tracing from where the diseased animal was originated. If possible, tracing back up to the herd level, unless at least we have to trace back from which district the infected or the CBPP infected animal was originated. And trace forward means if checking if another animal was transported with the CBPP infected animal, if so, where it was sold, which abattoir or which market bought that infected animal. And the final approach is the reporting and the recommendation. The role of participatory abattoir surveillance in control of CBPP: early detection of the disease. Just my time is running. I will. The other role is evaluating the control programs. Regular abattoir findings provide feedback on the effectiveness of vaccination campaigns and other interventions. If the post-mortem examination at the abattoir revealed or indicated lesions of CBPP from a district or from a place where CBPP control was implemented, it is an indication that the program is failing. Supplementing passive and active surveillance, it ensures continuous monitoring since slaughtering occurs year-round. Trade assurance, cost-effective and sustainable surveillance. We are not going to add any expense in order to conduct CBPP surveillance because we can use or we are going to use an existing water infrastructure and trained meat inspectors, so it makes inexpensive and cost-effective, especially in resource-limited African countries. The major findings of participatory abattoir surveillance, this is, I think, addressed by my colleagues. Why participatory abattoir surveillance is important? It provides a cost-effective and a continuous monitoring system, especially in regions where clinical surveillance and the laboratory infrastructure are weak. It measures the economic loss due to contamination, determines the spatial and the temporal distribution of diseases, estimates the agreement of diagnostic techniques. For example, the study conducted in North Ethiopia indicated at the abattoir 10 animals out of 192 animals were positive with CBPP. That means 5.2 in prevalency. Whereas by serology in the same place, it showed result 3.4. There is a gap between the findings. That indicates this indicates as Abattoir surveillance or Abattoir examination is an accurate approach to collect an accurate data. Uh, Limitations. It does not capture animals that do not enter the slaughter chain. Traders may sometimes misreport origin if they have no experience in such like they misreport the origin. To also requires skilled staff for leisure recognition in the abattoir, the CBPP leisure recognition. Some traceability depends on cooperation of traders. If there will be no traders with us, we will miss to trace the sample or the origin of the animal. As a recommendation, these are a lot of recommendations. Thank you, but I don't know how I finished 30 minutes. Sorry.
Thank you very much,
Hello.
Dr. Garoma Desa. I think I have pushed you a bit too hard, but it's okay. It leaves us with about 10 minutes for questions. Are there any questions from the audience? Maybe I have a question. Oh, Massimo has a question, and I have a question as well then. Massimo, go ahead.
Thank you very much. No, thank you, Garoma. Thank you for your presentation. I would like to know from you if you can summarize. We have 10 minutes, and Patrick have to also has a question for you. But if we can summarize, in your opinion, as epidemiologists, which role can play the abattoir, in your opinion? Because your presentation was very good, very high level. But for an old person like me, I would like to know, in your opinion, which approach we have to use using and the different abattoir that you can find in Africa, not only the official one, and which abattoir is better for you? Which one is the better one? The industrial one, the local one? Thank you very much.
The user one? Sorry, Massimo.
The industrial one, the big one, or the town, the small town abattoir, or the village abattoir? And which role can play the bush abattoir? This is my question.
Thank you.
Practically, what we can do, in your opinion, what we can do considering the different typology, the different kind of abattoir that we have in the African context? Thank you.
Thank you to Dr. Massimo. Thank you for your good question. As you raised, there are different types of avatars, but the bigger one is, as to me, the bigger one is a good approach because it is an avatar with good infrastructure, an avatar with good trained expertise, good trained veterinary expertise, meat inspectors. If we take another avatar at the village level, at the district level, or local avatar, they have no enough infrastructure. Even in some places, there are no enough lights. If the slaughter is going to be conducted in the night, even they have no enough light, they have no enough water to maintain the sanitation, to maintain the biosafety and the biosecurity, though the industrial one is, as to me, the industrial abattoir is preferable for me. Thank you.
Thank you.
Yeah, Mike. So at the last SGE meeting, we had some interesting presentations about participatory epidemiology in the detection of CBPP in the villages where the animals are raised. That was an example from Tanzania. My question was actually looking at the pictures that you showed of participatory epidemiology. I didn't see anything happening at the abattoir. So my question is, what exactly is the difference between, say, clinical surveillance by abattoir workers and inspection by abattoir inspectors? It seems to me that it's a very thin line.
Okay, thank you. As we know, contagious bovine pleuropneumonia by its nature is asymptomatic. Most of the time, it is asymptomatic, and it is also chronic. We can get less acute CBPP. So this makes the clinical surveillance difficult. It makes it is asymptomatic nature, and it is asymptomatic, and the chronic nature of the CBPP makes the clinical surveillance difficult. But in the abattoir, in the slaughter, though we cannot able to detect the disease at antemortem inspection, we can detect the CBPP lesions at postmortem inspection. So the clinical surveillance is difficult to get or to detect the disease, but at postmortem, at the abattoir surveillance, the postmortem inspection makes us it is a good tool to get data about the CBPP. May you get my idea?